Embryogenesis in monocots are a little different, particularly because monocots do not have cotyledons. The succeeding slides will illustrate the morphogenic events in monocots during embryogenesis. Monocot embryogenesis also starts with a zygote, which is the product of the fusion between the egg and the sperm cells during double fertilization. Like in dicots, the zygote divides to give rise to a two-cell embryo composed of an eggshell and a basal cell, as indicated by the letters A and B in this figure. The eggshell cell undergoes cell division and gives rise to a globular proembryo. The basal cell, on the other hand, does not divide but forms the terminal cell of the suspensor. Other suspensor cells are also produced by the eggshell cell along with the embryo. The globular proembryo then divides and elongates into a club-like shape. This is the early coleoptile stage, and as the name suggests, this is when the formation of the early coleoptile is initiated. The coleoptile is the protective sheath covering the first true leaf of monocot plants, such as grasses and cereals. In this example in the left, we have the coleoptile of corn as indicated by the black arrows. Maybe it's clearer in this picture here. In the right, we have another example of how a coleoptile looks like in rice. The coleoptiles are the ones indicated by the white arrows here and here. Further cell division of the embryo at the early coleoptile stage moves the embryo into the late coleoptile stage when the shoot meristem becomes visible. Shoot meristem here is abbreviated as SM. Then we have the first leaf stage, which is characterized by the differentiation of other tissues, including the scutellum and the radicle. The scutellum is abbreviated as SC, and the radicle is abbreviated as RD. The scutellum is the equivalent of a modified cotyledon in dicots, while the radicle is just another term for embryonic root. In this right panel, we have here a seed with a small structure protruding from it. That is actually the radicle. As the seed becomes autotrophic, the radicle develops into a complete root system, just like what you can see here. Following the first leaf stage is the second leaf stage. This stage is characterized by more advanced differentiation of the first leaf and the initiation of the second leaf. Then finally, we have the mature embryo stage. During this stage, all tissues are evident, with the addition of a third leaf, shoot meristem, root meristem, and the coleorhiza. The coleorhiza is a protective sheath covering the root of a germinating monocot. After the embryo matures, just like in dicots, the mature embryo will enter post-embryonic development via a controlled process of desiccation. This drying event will put metabolic processes in the embryo in temporary arrest or suspension, effectively putting the embryo into a dormant state. To summarize, after double fertilization, the zygote and primary endosperm will undergo several rounds of cell division to develop into a mature embryo and endosperm. Embryogenesis refers to the process of embryo formation and development from the zygote. This process can be divided into several morphogenic events, namely morphogenesis, embryo maturation, and desiccation. The early rounds of cell division of the zygote gives polarity to the embryo. Embryogenesis in dicots and monocots differ in terms of structures that are formed. In dicots, embryogenesis produces structures like the hypophysis and suspensor. In monocots, embryogenesis gives rise to specialized structures not found in dicots, such as coleoptile, scutellum, and coleorhiza.